So next for session 2 is Shwarya and Don. Why don't you tell us what transpired and what lessons we can take away?
Great, thank you. And I want to thank Dr. Subbiah and Drs. Bertinoli and Singh for the partnership on this. This session was a pleasure to put together and certainly to think about from our perspective. So as we had all of our excellent speakers go through and think carefully about each phase of early drug development as it pertains to the older adult population, we really had to start at the laboratory level and the non-clinical data sets. And so the non-clinical data is woefully inadequate when we start to think about translation of information from that data set to the older adult population. We have targets, we have pharmacodynamic targets, and certainly a lot of cell line and animal data, but that data doesn't reflect and is often done in very young animals overall and doesn't reflect the geroscience that's known. Secondly is that our phase 1 trial designs, whether they be first in human or combinatorial phase 1 studies, are just not inherently amenable by their current construct to older adult enrollment. We have many days that are long for patients. We have frequent visits within a 3 or 4 week period, particularly in cycle 1. Those can be challenging for older adults to tolerate and sit through. We may have limited sites for those studies. Out-of-town travel and cost that goes along with participation in these studies, overnight stays, other aspects of costs, pre-trial screening procedures, which can be invasive and frequent, not only biopsies before study, but also on study, as well as safety assessments such as cardiovascular function, ocular examinations, and others. Third, really endpoints that matter to older adults. We all focus by the old construct of phase 1 studies that we're very concerned about a grade 4 thrombocytopenia in cycle 1, for example. Most patients, and particularly older patients, really don't care about that. They care more about grade 1 nausea that lasts 4 or 5 months, or diarrhea that doesn't allow them to travel or to go to the movies. And so I think all patients really care more about that, but particularly the older population is an important aspect of that. And then quality of life and functional impact. And I thought there were great discussions in our session about overlapping toxicities, things that may be additive that we don't think about. 1 + 1 can equal 4 sometimes when it comes to grade 1 toxicities. If you have multiple grade 1s, we have to remember that those can be problematic and as problematic for patients as we go deeper into study. Eligibility criteria has been mentioned already, but I believe that we've missed opportunities within phase 1 studies where we have opportunities to get safety signals that we can learn from early in older adults and more frail populations that can be managed. We are, I think, overly restrictive sometimes in the way that we think about safety signals, even with first in human trials, and exclude relevant populations. And more specifically, we need to do a better job, or the issues that we're seeing is safety data that really is not rapidly incorporated into the next phase of study or even within the same study, where we can start to learn more about eligibility criteria. And finally, really a great talk on stats and that lack of wider adoption of novel designs of phase 1 trials really still continues to be a 3+3 world that we live in. And we've got a lot more tools in the toolbox that we're just not utilizing enough. So next slide, move into the solutions. And the first one I think I alluded to is moving a lot of the geroscience that's out there into the non-clinical setting. And so really focus that geroscience on genetic abnormalities, epigenetic changes, not only for safety, but potentially, as was mentioned in another session, potential differential in activity in a positive way. We oftentimes worry most about toxicity, but we can't forget that there might be instances where drugs may be more effective in an older population than younger. Really expand the trial site and the definition of a site. Not only those phase 1 centers or early phase trial centers that we all know of, but also local sites, local laboratories, telemedicine can probably be utilized more than it has been. Use web-based apps. We certainly have populations of older adults that are very tech savvy, and when possible, using those applications for patient-reported outcomes and others early on can be really helpful. And finally, an area that I'm interested in is try to use more data modeling and in silico modeling to extend and expand PK and PD collection in a way that doesn't require multiple blood sets. And so try to make sure that we're making that patient friendly that it's not just the pharmacokineticist that's making us get these 12-hour PK values on 7 different days in a given cycle. And so use dose derivation models. I think that's an important point, is that we don't need to have a one-size-fits-all dose strategy. And we've seen that on many occasions, that a dose should not be a 0 or 1 variable. It does need to be something that's got ability to change throughout a population within a label based on tolerability as well as efficacy. And so using some of the other designs that are out there, perpetual designs, where you might slot in frailty measures as a bucket of patients to define dose, consider perhaps intra-patient dose escalation in a frail population where a patient goes up on the dose cycle to cycle to see what their tolerability might be, or a population's tolerability might be. And then think about continuing toxicity monitoring, again, moving beyond cycle 1 to assess tolerability of grade 1 and 2 adverse events that might be more perpetual. And then again, think also about differing doses. We need to use the more extensive Bayesian designs to ensure that we get more data at dose bands that are that's robust and that we can use across areas. And all this is based upon the idea that we've opened the gates more to older adults and frail populations. And then I think an incredibly important point is to make it as easy as possible for all of our patients on trials, but particularly our older adult patients, by providing overnight accommodations, much like was mentioned, the Ronald McDonald House for kids and others. We need to think carefully about allowing and creating those spaces for older adults that might have specific features and resources for those patients. The next slide. And then we'll talk a lot, people talked a lot about eligibility criteria, so I won't go into this too much, but we certainly need to expand them as early as possible within a given safety set. It still baffles my mind that we might have a novel monoclonal antibody that's very targeted, that we can predict what's the likely adverse event profile, and yet we mandate that patients have to have a creatinine clearance of 60 or higher. That doesn't make any sense from a pharmacokinetic and pharmacology perspective, and rarely makes sense from an adverse event perspective. And so, even just moving that needle would start to change the number of patients that could be eligible just from renal function. There are other aspects, but I think those are important things to consider. We need to think about learning trials early on in early phase studies, as well as think about outcome adoption and enrollment as we go on into studies and drug development. We need to really incentivize physicians to refer older adults to early phase trials. That has been mentioned as a gap and a barrier repeatedly, and we've got to create an incentive package for all community oncologists and academic oncologists to refer those patients that are older to these early phase trials. And remember that we have drugs coming forward that have wider therapeutic indices, and eventually these drugs are going to be used in the older adults. So why not get early data as much as we can? And then finally, simplify, simplify, simplify. And that's informed consents, that's visits, that's correlative study schedules, and other things, so that it's easy not only for the older adults, but also for other patients and populations. If you don't need it, don't get it. And that is often I think an overlooked aspect of some of the studies that we do. And we have the holy grail of biopsy data and other things. And, that's a whole separate workshop on how we really don't get the data we need from biopsy. So we need to think more carefully about the ethics of doing them. So with that, I'll stop, and we'll move on to our next session. Again, I want to thank Dr. Subbiah for her partnership in this session.
Yeah, that's terrific. Really an incredible summary of that session.